translation function of stratification of cancer drugs to integrated drugs. Similarity.
So, good morning, everyone. My name is Sina, and I'm currently doing my master's studies here in Istanbul at Koç University. So, let's jump to the first slide. So, the intertumor and intratumor heterogeneity of cancer is known. And we can also observe the modulation of drugs and diseases in their signaling pathways. And finally, we can measure the interplay between multiomic molecules. So, with this, we will try to understand the drug action mechanisms and also realize how to classify drugs in a condition-based manner. So, drugs perturb simultaneously a set of protein networks beside their immediate targets. For example, here, we can see that these two drugs, with the same target, they modulated separated networks, while these other two drugs, with the separated targets, they have the shared modulated networks. This shows us that with a network-based approach, we may reveal unknowns about drug response, and also we can predict effective drug combinations. So the main idea here in this study is to transform the input omic data into clinically interpretable knowledge. So to do this, we collected transcriptomic and phosphoproteomic data of 5 cancer cell lines treated with 70 drugs from Seyma, and we found the upstream regulators, the transcription factors of these significantly expressed genes from this data. And finally, we also collected the targetome data from Seyma. We merged this data together to have a seed proteins list, including drug targets, phosphoproteins, and transcription factors, to construct our disease cell line pair modules. So here, as I said before, with a multiomic integrative approach based on reverse engineering principles, we reconstructed drug cell line specific subnetworks. The idea in the network approach is to first find unrecognized pathways from the initial data and then find hidden proteins in those pathways. To do this, there are several tools available developed from our group, which we used omics integrator here in our study. And finally, we become able to reconstruct around 200 drug cell line pair subnetworks. And we applied topology separation on these subnetworks. So with our subnetworks, we calculated all pairwise separations and come up with a separation score matrix here. For example, you see the separation score for a specific cell line here for their different drugs. Here in the bottom, you see the separation scores we calculated, and the top triangle shows the cell line and drug pair types. For example, if the drugs are from different mechanism of actions or from the same mechanism of actions, which I can explain later. So our first finding here was that the chemically and functionally different drugs may modulate overlapping networks. For example, here, these two drugs, although they have different protein targets, but they modulated the same networks, as I showed earlier. And you can see the common proteins in a purple color here. And we also validated from the literature data mining and find that these two drugs, although they have different targets, but they share several downstream transcription factors. Our second finding was that the topologically separated drug networks may guide the use of drug combinations. For example, here for two other drugs, their networks only share 3 proteins here, but one of these proteins is a cancer driver gene, BRAF. And also, we found the synergistic effect between 2 drugs. And with this approach, we actually collected around 1,000 drug combination proposed here for the synergistic effect, which of course should be validated in future studies. So the part so far was actually published in Nature Systems Biology, and the ongoing part of the study is when we realized that the topological separation between the drugs can give us clues about their sensitivity levels. And so another use of drug network here is to understand the drug resistance mechanisms. And we observed from our networks that cell lines networks that are perturbed by the same drug and have a different response, they have little topological similarity. But to do further research, further analysis, we had a big limitation in data of sensitivity, and there was no sensitivity and resistance clearly separation in our subnetworks. So we actually decided to reconstruct our networks with another approach. So here, instead of using treated cell lines, we actually used non-treated, non-perturbated cell line data, which are more abundant. We started with 959 cell lines, and we reconstructed about 28,000 subnetworks, from which around 1,000 of them are clearly separated as sensitive and resistant. And with using these as labels, and with a graph embedding method followed by a machine learning approach, we become able to learn and predict and classify actually the resistance of these subnetworks treated by drugs. And actually we simulated the drug effect in our analysis with a propagation method here. And this is the method, and our predictions are validated with 10-fold cross validations, and you can see the results, the accuracy and AUC values here, as well the ROC curve. And finally, to summarize, I can say that network models can identify common or context-specific pathway level changes. Reverse engineering from initial omic data to context-specific subnetworks can shed light into network level change to observe hidden commonalities. And finally, networks are useful in revealing potential similarities or combination between drugs that cannot be identified from their structural similarity at first. And finally, I would like to thank my advisor and my colleagues in our network modeling research group, and especially Seyma, who was my colleague in this research. And I would like to thank RECOMB Technologies for their support in RECOMB conference registration. And I would like to thank you for listening.
Thanks. So you showed this drug-drug similarity matrix, right, which uses this model-based similarity method. And I assume you're not just including cancer drugs, but also all types of drugs, right? Or is it specific to cancer drugs?
It's specific to cancer.
Okay. Anyway, if you would do some sort of hierarchical clustering on this similarity matrix, do you find the drugs together that target the same cancer types, or does it somehow make sense?
Actually, we did that, and it's also included in our matrix, in our data, which is available in the paper.
Okay, and does it make sense? So the clustering, I mean, do you see the drugs that target?
Yeah. Actually, I thought about this question, and this is the complete matrix of our all data here. And we did the hierarchical clustering here and sorted our drugs in that manner. And we actually, in the bottom part, we saw the separation scores, and in the upper part, we can see that if that drugs do share common pathways and share mechanism of actions, going to